Hello, my name is Igor Gago, and in this video I will be presenting my master thesis project. Mars is a planet of great astrobiological interest to scientists, with numerous past, current and future missions dedicated to this same field. These missions include, for example, the Perseverance rover, which arrived on the Red Planet on the 18th of February of 2021, with the main goal of searching for signs of past microbial life and past habitable conditions by identifying and collecting the most compelling rock core and soil samples, which could be brought to Earth on a future mission for more detailed study. Another example is the ExoMars 2022 mission, which will be launched in 2022 and has an estimated target arrival of June of 2023 and will deliver the rover Rosalind Franklin to the surface of Mars. This mission has a primary objective of studying an ancient site at Mars interpreted to have strong potential for preserving physical and chemical biosignatures, especially from the very early history of the planet. The surface of Mars is a highly inhospitable environment, characterized by extremely low temperatures, barren conditions, high levels of ionizing radiation, and an absence of liquid water that inhibits almost any Earth organism from surviving without some extreme adaptations. And yet, the search for life on the Red Planet is a highly demanded field nowadays. The characterization of the Martian radiation environment and the effects of this same radiation on biological matter are essential to understand if the Red Planet can sustain life or if it has fostered life in the past. DNA is the molecule carrying genetic information for the development, functioning, growth, and reproduction of all known organisms. So, if we learn more about the possible hazardous effects of radiation at the DNA scale, we can obtain relevant information about the possible sustainability of life on the subsurface of Mars. Before studying the effects of radiation on DNA molecules, it is important to know which Martian sites have high biosignature preservation potentials. That is, which locations in the Martian subsurface have conditions more propitious to the fostering of life and the preservation of past extinct life. Research shows that Earth-like habitable environments in the subsurface of Mars could exist wherever groundwater is present. In this case, locations with subsoils rich in sulfides could harbor sulfate-reducing bacteria, just like on Earth's deep continental subsurface. Research also shows that when it comes to Martian sites, both on the surface and the subsurface, soil compositions with a higher percentage of hydrogen have lower radiation doses due to the reduction of the albedo nutrient flux, and thus are able to shield more efficiently the potential subsurface organisms. My master thesis project is based on two main steps. First, modeling the linear energy transfer spectrum of the ionizing field created by incident cosmic rays and solar energetic particles in water at different depths at the Mars subsoil up to 3 meters, using the detailed Martian energetic radiation environment model. And second, simulate the early radiation-induced damage on a simplified model of the DNA molecule due to the most significant components of galactic cosmic radiation, using the Gen4 DNA extension. The main contribution that my thesis project brings to the literature is its interdisciplinary perspective, by bringing together physics and biology, all while utilizing stochastic Monte Carlo techniques to provide highly relevant information about the possibility of current or past life on Mars.